Okay, so today we are going to go over the light-independent reaction. So last time we went over all of the photosystems, photosystem 2, which happened first, photosystem 1, which happened second. And then today we're going to talk about where those products go. Obviously, we already talked about whenever water is split in photosystem 2, the oxygen part of that goes away out of the stomata, and we breathe that in. But we didn't talk about the ATP. We didn't talk about the NADPH, where it goes after it's formed. So your book goes over just a comparison between the chloroplast and the mitochondria, just so you guys know. We are going to, in the mitochondria, transfer chemical energy into ATP. And in the chloroplast, we are going to transfer light energy into ATP, but not a lot of ATP, not as much as we get into the mitochondria, which again, as we talked about last time, why there is a need for both the chloroplast and the mitochondria in plants. Okay. So we already talked about the protons being pumped in the chloroplast. They go to ATP synthase, and that's how we are going to make our ATP. Now, whenever this process happened, we had the electrons kind of being around that thylakoid membrane. And on one side, we had the inside of the thylakoid. On the other side, we had the stroma. Now, the Kelvin cycle, which is our next thing, is going to happen in the stroma. So if we formed our NADPH and our ATP inside of the thylakoid, all that would do is make it for a longer journey for the NADPH and the ATP to get out of the thylakoid. So it'd have to go through that membrane again somehow, go to the stroma, then be used by the Kelvin cycle because, as we just said, the Kelvin cycle occurs in the stroma. So this is why ATP and NADPH are actually formed on the side facing the stroma instead of the side inside the thylakoid. It's less of a journey for those two molecules. And here was the picture we went over last time, guys. Okay, so the ATP and NADPH are now going to go over into the Kelvin cycle. Again, that occurs in the stroma. All right, and here's even a better picture of where the Kelvin cycle is going to happen in the stroma. Okay, and you can see the ATP and the NADPH are going that way. So let's start on our Kelvin cycle. All right, the citric acid cycle is a catabolic bunch of reactions from our cellular respiration, meaning catabolic, we're breaking it down. The Kelvin cycle, on the other hand, is going to be anabolic. We actually build things up, okay? So I'm going to draw a picture of the whole Kelvin cycle for you guys, but we are going to be taking in carbon dioxide for this part, okay? We're taking in carbon dioxide. And what we're going to do is we are going to use the carbon dioxide to form sugar. Now, the sugar that we are originally forming is not going to be glucose as we know it. Instead, it's going to be something called G3P, which is sugar, but it doesn't actually make the full sugar molecule right away. Okay? So our first phase is we are going to go through something called carbon fixation, which is where we are going to bring in our CO2, we're going to use it, and we are going to combine it with something called RuBP. And what RuBP is rubisco 1,5 biphosphate, okay? Meaning we have rubisco, and then attached to each side of rubisco, we are going to have phosphates. And rubisco is going to have 5 carbons to it. So once we combine it with our carbon dioxide, we are going to get 6. All right? All right. So let's get into it. All right. So we're going to bring in some carbon dioxide here. Okay. Oh, I do not like using gray. Let's use black. There we go. So we're going to bring in some carbon dioxide. Okay. Now again, this is a cycle just like the citric acid cycle or the Krebs cycle we went over last chapter. All right. I'm going to bring in 6 CO2s. And if you remember from our photosynthesis equation, we did have 6 CO2s we had to bring in order to form our C6H12O6. Okay. Now, from the cycle last time it went through, we are going to have our RuBP. That's going to combine with our 6 CO2s, our 6 carbon dioxides. Okay. And if I wanted to draw what RuBP looks like, each one of them, we would have 1, 2, 3, 4, 5 carbons. And on each side of that carbon, we are going to have a phosphate attached. Okay. So that is what our rubisco looks like. It's a 5-carbon molecule. Okay. Now, each time I draw an arrow, guys, yeah, an enzyme is going to be present there in order to catalyze that reaction. In this one, we're going to have a rubisco enzyme. Okay. And when we put these together, we are going to get some 6-carbon unstable molecule. All right, it is unstable, and I'm not going to worry about drawing it there because it's not important to us. After we do that, all right, we are going to get something called G3P. Okay. How many are we going to get? We're going to get 12 of them. And each one of the G3Ps look like this. We're going to have 3 carbons attached to a phosphate here. Okay. Now, there are other things around the outside, guys, that I am not mentioning. Okay. We don't need to know them, but we will be changing some things shortly with it. So along with this, in order for whatever the 6-carbon one here was, guys, so we had 6 carbons with 2 phosphates attached to the side. Again, it's unstable. So what we're going to do is we're going to bring in 12 ATPs all right, and we are going to take that, use that energy, and it now becomes ADP. Okay. What we're going to do is use that energy. It's going to break our molecule apart. Along with this, we are going to use our NADPH. How many did we have? We're going to have 12 of them. We're going to take that down to NADP+. Okay? And that's our electron donor so that we can break that 6-carbon molecule apart that is up here. Okay. Next step. After we have that, that's a G, guys. I'm sorry. It's called P-gal. All right. We are going to have 10 P-gal left. Now, P-gal, C, and I'm going to come back to this step here, guys. Oh, I drew my carbons the wrong color. Let's keep them consistent. Let's go back to red. Okay, we're going to have our CCC and a phosphate attached to it. Now, for P-gal, the things on the outside are going to be a little bit different than our things on the outside of our carbon. Okay, I'm not going to worry about drawing that in there. But if you notice, we have 12 G3Ps over here. Each one of those G3Ps have 3 carbons to them. So 12 times 3, we have 36 carbons. Over here, we have 10 times 3, that's 30. Where did those other 6 carbons go? Well, they exited our Kelvin cycle, which I probably should write. This is the Kelvin cycle up here. Those other 2 P-gals, or G3Ps, whatever you want to call them, they're going to exit. Again, they're both with 3 carbons. They're going to go through some different chemical reactions, and they are going to, in the end, form our C6, our H12O6. And that's our goal of the Kelvin cycle, okay? So our goal is to form our glucose here. But even though we are done, we formed our correct product that can now go to the mitochondria and make a whole lot of ATP, or fuel a lot of ATP, we got to make sure that this P-gal can get back and form RuBP again. Because without it going back to that, the cycle stops. There's no more RuBP. We cannot combine that with our carbon dioxide, which means later on we will not get any more glucose. So we want to make sure that we can complete this cycle here. So what we're going to do, again, this is an anabolic process and not a catabolic like in the Krebs cycle. We are going to transform these a little bit. And we are going to get 1, 2, 3, 4, 5 carbons attached to a phosphate. Okay. How many of them? Well, 1, 2, 3, 4. I got 5 carbons there. I'm not losing any carbons here, guys. We got to keep everything consistent. So 10 times 3, I got 30 carbons. 1, 2, 3, 4, 5. What number times 5 will give me 30? That's going to be 6. So I'm going to have 6 of this intermediate molecule here. And then lastly, in order to stabilize it again, and again, I'm not going over all the unstable ones here with you guys. We are going to, once again, bring in some ATP. How many? Whoa, why did I draw? How many are we going to need? Well, we got 6 of these molecules, so I'm going to need 6 ATPs. And we are going to use those ATPs. And we're going to get 6 ADPs out of it, plus 6 phosphates. Those 6 phosphates are going to go to the other side there. And that's how we are going to get to our RuBP again. And then the cycle will continue. Okay? So that is your whole Kelvin cycle, guys. All right? Not the best drawing in the world, but you guys get the concept. I tried to make it as neat as I could. The goal, form our C6H12O6. Okay? Yeah, in doing so, I'm using a whole lot of ATP. I'm using it on both sides here, and I'm using all my NADPH from the photosystems. Okay? But I'm getting my glucose out of it, and that glucose can go to the mitochondria and form a whole heck more of ATP than we just show here. All right? So there is your Kelvin cycle. Here is the book's version of it, guys. They go through some of the intermediates. I'm not going to, like I said, I'm not going to go through all the intermediates with you guys. All right. So we went through all the 3 phases, and we got our RuBP back, so we're done there. Now we're going to talk about different types of plants that maybe can't go through the Kelvin cycle for one reason or another. The first one we're going to talk about is photorespiration. Now, I mentioned the rubisco enzyme, which is going to combine with our carbon dioxide to form our 6 carbon molecule. So we did the RuBP plus the CO2, gave us a 6 carbon molecule. All right, there is an enzyme, the rubisco enzyme, that is going to catalyze that reaction. That's going to bring the carbon dioxide with the RuBP together. Okay. How does the carbon dioxide get to the Kelvin cycle? Well, very simply, the plant opens up its stomata. When the stomata is open, that stomata will bring in the carbon dioxide, and the carbon dioxide will go to the Kelvin cycle. Easy enough. Okay. However, as the plant opens its stomata, the problem is if the stomata is open, the plant can also lose water, which we don't want, especially on hot days. Okay, it's very easy for the water to just leak out of the plant, evaporate, that type of thing. Okay, without water, we can't run the photosystems. If it's a really hot day outside This means that we cannot bring carbon dioxide in, and we want a lot of carbon dioxide in. Whenever we do photosystem number 2, we want that oxygen to almost leave immediately. The plant does not want that oxygen inside of it. If the plant cannot open up its stomata, it's going to go through something called photorespiration. I'm going to give you a picture of photorespiration. We're going to draw that out, and I'm going to show you that process. So, we are going to, let me see if I can, we're going to bring this up here. Here is photorespiration, and I'm going to make it very simple for you guys. Okay. So we have our RuBP from the beginning. This is going to be photorespiration. We have our RuBP. Right here, draw it in blue, we have our rubisco enzyme. Probably something that ends in ASE. We're not too worried about it. Okay. Here's the problem. We cannot bring in CO2 because our stomata is closed. The other problem, you would think, okay, no big deal. We can just, wait to go through the Kelvin cycle for whenever we do have CO2 present. Remember, guys, none of this stuff has eyes. They can't see things. The problem is the rubisco enzyme does not just have an affinity for our carbon dioxide. It can bind other molecules other than carbon dioxide. One being, guess what? Oxygen. Okay. So in this case, our rubisco enzyme, if I draw it here, we'll just draw it with 2 spikes, I don't know. Okay. Along with it being able to bind, let's say this is carbon dioxide here. For some reason, it can also bind oxygen. This is why we want oxygen to leave as soon as possible so that it does not bind, okay? It does not bind with our rubisco enzyme and that carbon dioxide can get in there. But if oxygen can't leave, it's going to bind, all right? So here's what happens. So I'm going to erase some of this stuff over here. We need to do a cycle again. All right. With this, oxygen is going to combine with our RuBP, and it's essentially still RuBP. It's still a 5-carbon molecule. All right. Well, that's not good. Okay. So now, what we're going to do is we are going to have to take, we're going to get 6 PGAs out of this, okay, which was our 3-carbon molecule. And we are going to add that to 6 2-carbon molecules. Okay. Those 2-carbon molecules are called phosphoglycylate. So you don't have to know that. What we're going to do is we are going to take these, okay, photoglycolates. We are going to take them to the peroxisome, which is another organelle. All right. And it's going to be changed into something called glycine, where you don't have to worry about these, but the peroxisome is then going to ship it to the mitochondria. Whoa. Sorry guys, I ran out of space there. Peroxisome ships to the mitochondria. The mitochondria, so this is number 1 here, it goes to the peroxisome, 1 organelle. Gets changed there. It goes to the mitochondria, which is our 2nd organelle. The mitochondria is going to take it. The peroxisome makes glycine, okay? The mitochondria is going to take the glycine. It's going to transform it into serine. The mitochondria is then going to take it back, I know, to the mitochondria. Crazy, right? So it's going to go, I'm sorry, back to the peroxisome. It's going to go back to the peroxisome. Peroxisome can then take the serine in and transform it into glycerate. Then, after that, what can happen is that peroxisome can make CO2, given that, okay? And then it can go back to our PGA and continue through the cycle, and we can still get our C6H12O6 out of it, okay? But the whole thing I want you guys to get is photorespiration, yeah, sure, the plant can still do it. It can still go through the light reactions because it just needs sunlight for it. But the issue here is that without the carbon dioxide just breathing in through the cell, it has to form its own, okay? I mean, think of it with you guys. Wouldn't you rather just be given something than, oh my goodness, I got to form my own stuff, you know? The downside of this is it takes a lot more energy for these things to go through photorespiration for the plants instead of it just having that carbon dioxide, all right? Will the system still complete itself? Yeah, it will. It's just, again, a lot more energy. So photorespiration, yeah, it's a good backup plan, okay? But you're going to have to go through the photosystems a lot more, create a whole lot more energy in order for that to occur, all right? Okay, guys, so we went through photorespiration. We're good there, all right? Again, it consumes a lot of ATP. We use oxygen for it. The rubisco enzyme has a good affinity for the oxygen, all right? All right, so we are good. We're good here, guys. All right. The one thing, the first part with here, whenever the rubisco enzyme was probably first, whenever it was evolving, whatever, there probably wasn't a lot of oxygen in the world because oxygen levels didn't start to like escalate or become plentiful until the plants did a whole lot of photosynthesis. So at first, it probably developed and it was just like, oh, look, carbon dioxide's everywhere. And it didn't have to worry about having that affinity for oxygen. But as the levels build up, photorespiration just became a natural evolutionary thing because of that affinity. All right, guys, next time we'll go over C4 plants and CAM plants. I think this is enough for today for you guys to chew on. Have a good rest of your day.